Why does fufu cook faster when you add a special leaf? Tiny helpers called enzymes speed up reactions in your body too. Today we explore enzymology. An enzyme is a biological catalyst, usually a protein. It speeds up chemical reactions in living cells, but is not used up itself. Think of it as a tireless worker. Enzymes have 4 key properties. They are highly specific, reusable, very efficient, and sensitive to temperature and pH conditions around them. Most enzymes end in ase added to their substrate name. Sucrose breaks sucrose. Enzymes fall into 6 classes based on the reaction type they catalyze. In the lock and key model, the substrate fits exactly into the enzyme's active site, like a key into a lock. Only the right shape works. The induced fit model is more accurate. The active site changes shape slightly to grip the substrate, like your hand molding around a mango. Some enzymes need helpers called cofactors. Inorganic ions like magnesium or organic coenzymes like NAD activate the enzyme. A poenzyme plus cofactor equals holoenzyme. Activity rises with temperature until about 37 degrees. Then enzymes denature and rate crashes. Each enzyme also has an optimum pH where it works best. As substrate increases, rate rises, then levels off when every active site is busy, but adding more enzyme increases rate steadily, since more sites become available. Many students confuse denaturation with inhibition. Denaturation permanently destroys the active site shape from heat or extreme pH. Inhibition is reversible, blocking by another molecule. Remember enzymes with the word SC in TE: specific, catalysts, efficient, not consumed, temperature sensitive. Picture a sweet scent guiding substrates to the active site. Subscribe to Ovasaabi for more biology lessons. Visit ovasaabi.com to continue your journey to exam success.